my mission for the last 2 years is to try to figure out how our genetics play a role when expressed with our cannabinoid journey.
Welcome to the Greener Grass podcast from Bluebird Botanicals. I'm your host, Lex Pelger. Len May can read your genes and match you up to the best cannabis cultivar for you. Which ones might cause you to feel anxious or relaxed? Which ones will wake you up or knock you out? Lots of people just go for Indica or Sativa, but that dichotomy is an outdated and blunt tool. With Len's research at Endocanna Health, he's uncovering a much more complex chemical interplay between our bodies and our plants. Today we'll explore this brave new world with our guest, the pioneering activist for cannabis, Len May. This show is brought to you by Bluebird Botanicals, to spread education about cannabis and other things on the greener side of life. Bluebird CBD oil comes from farms in southern Colorado and is grown using only water, soil, and sunlight. Go to bluebirdbotanicals.com for more info. Hello, everybody. I'm very pleased to be here with Len May, who is, among many other things, the CEO of Endocanna Health. Thanks for coming on the show.
Thanks for having me. Appreciate it.
And before we get to your most recent venture with Endocanna, when was it that you got interested in cannabis?
Well, I would say I got interested in cannabis as a teenager. I was diagnosed with ADD when I was in my early teens, and I was put on all kinds of prescription medicine, and most of it didn't work or made me feel bad. So I accidentally discovered cannabis hanging out with some older kids, and I thought that we're going to smoke a cigarette. And they gave me what I thought was a cigarette, and I took a puff of that, and ended up not being a cigarette. So when I went back to class at that time, uh, the world slowed down for me, and I was able to focus because my type of ADD is sort of like a dreamer state ADD. So I have a lot of bunch of different thoughts that pop in my head, and then it's hard for me to concentrate, or was at that time. So this was, yeah, I would say a very long time ago, and maybe over 30 years ago, and then so it became my go-to medicine.
And so then how did you continue your pursuit of cannabis and make it into a life's work?
Yeah, so I became an activist, and I held a rally in Philadelphia, where I'm originally from, in at Independence Hall, where the Constitution and Declaration of Independence are both located. Uh, I should mention that both are on hemp paper. Uh,
Amen.
my keynote speaker was this lady named Elvie Musica, and she was one of the first people to get cannabis from the U.S. government under the IND program for her degenerative glaucoma. And she spoke at this rally, and I was, like I said, the president of this Cannabis Action Network, and I became a lifelong activist and really pursuing this as my life's work and life's journey. So I try to figure out a way how I can get into the space besides just trying to help people with different chemovars. So when I moved to Los Angeles, I get involved in the dispensary space. So I was involved in a number of dispensaries and the cultivation facility, and I became sort of obsessed with that. And the one thing that was really frustrating to me is that people with different symptoms, different conditions, or whatever they wanted to achieve, they would look for a certain strain or chemovar. And after that, we would say, okay, it's working for some people, it's not working for other people, or the strain wasn't right. So I really started diving deeper into the genetics. So the genetics of the actual plant itself. And I worked with a company called Medicinal Genomics. The first person genetically sequenced cannabis was this gentleman named Kevin McKernan. And I worked with him. And basically, what I did in the company, I traveled around the country and I would extract DNA from the plant, purify the DNA, and send it to a sequencer. So we could start trying to match up what the different strains really are genetically. And what interested me the most is that I could see that the genetic profiles have a different effect on different people. And that's really where it was a light bulb moment for me. I started realizing that we as individuals have a different response to cannabinoids and terpenes based on our own individual genetics and how they interact with the plant genetics.
Oh, that must have been a fascinating time. What was it like to be there watching the birth of the DNA of cannabis finally coming to light?
Yeah, I mean, it was an incredible opportunity to see how how off everybody is. So whatever names you call a certain strain in a dispensary, it doesn't mean anything because your profiles are really close. There's some parents, and then from there you have children of those parents, and they're very similar genetically. So all these names really don't mean much. The genetic profile of those really means a lot. But the thing that really fascinated me most of all is when I saw a research paper, an essay written by some of the people that worked for Cortigen Life Sciences, which was the parent company of medicinal genomics. And the paper that was written was specifically about epilepsy, or even specifically Dravet syndrome, a form of epilepsy. And that is the basis, a lot of it is the basis for the study that GW Pharma did with their Epidiolex product. And when I started seeing that, it was an amazing experience to see, okay, now we understand that genetics play a role in somebody's condition or symptom. Genetics also play a role in your cannabinoids and your experience. So, how do you match those two together? What if one person experiences anxiety, another person doesn't from the same exact chemovar? How does that work? So that became my mission for the last 2 years, is to work with our scientists to try to figure out how our genetics play a role when expressed with our cannabinoid journey.
And it's such a fascinating field of research. There's always more genetic associations coming out. And it's interesting, your company, not only do you have your own test strips, you can also accept data from any of the other major testing corporations. That must make things easier to get people into this endocannabinoid personalized health regime.
Yeah, absolutely. There's over 20 million people who have already taken a 23andMe, Ancestry, and many of the other genetic tests. So what we did, we build basically a translational engine. So we look for specific genes that are directly or indirectly, they have an effect on your endocannabinoid system. So we identified those genes, and then you can upload your raw data from any of the other sources that you already mentioned, and you can upload and get a report almost instantaneously. Now, the issue with that is that we're dependent on the genes that are tested under the version of the array that 23andMe or Ancestry or whatever use. So we will look at all those, and whatever ones are not being flagged by by those companies, we'll let you know that that data is not available, but it is available in our full advanced genetic kit.
For the nuts and bolts of it, how does the test help with recommending terpene and cannabinoid profiles for people?
Yeah. So, and I'm going to also answer a little bit longer the question that you said. One of the things you mentioned is this ever changing field. And you're absolutely right. I mean, we're only as good as the research that's available in the field. So we urge, science to continue doing research, and we build an AI algorithm that actually goes out with with some intelligence built in, and looks at different studies, different assays, different peer-reviewed assays and clinical trials, and sees how if it makes sense for us to add those into our research. So we're always looking to add. So if somebody's a member of Endocanna and there's new data about something else that came out, you'll get an update for life. So that's the way we always keep up to date with the latest and greatest data. But the way that it works is, I like to use some examples. So I just did a show recently with some athletes, and one of the things that we talked about was propensity towards anxiety. So if you look to see if you have a gene, if you have a genetic expression, there's a gene. Let's say there's a gene called COMT. They call it the warrior gene. So depending on where you fall on that spectrum, you can be a person who their fight or flight gets triggered really fast. You're an excitable person. You have a lot of dopamine and serotonin that's pumped in your bloodstream based on different conditions. Or you could be somebody who's more of a glass half empty, more on the depressive side. So your cannabinoid and terpene profile can actually work with you or can work against you. So if you're somebody that's more on the depressive side and you're consuming THC that has a lot of dominant myrcene, which is a terpene, which is basically a glandular secretion of the plant or essential oil. That terpene, myrcene, has an effect synergistically the way it works with cannabinoids. And if people are consuming that type of ratio and they're already on the depressive, in a depressive state, it may trigger further depression all the way to a psychotic episode if that gene is expressed. On the other flip side of that, if you're a person that gets triggered really fast with fight or flight and you have a lot of serotonin and dopamine that's pumping into your bloodstream and people consume we'll call it sativa dominant hybrids, but a ratio that's high in THC and limonene, which is a terpene that's prevalent in a lot of the sativa dominant hybrids. The limonene is also associated with pumping extra serotonin into your bloodstream. So if you're prone to anxiety, it will exacerbate or may exacerbate anxiety. So now this gives you a guide of a formulation ratio that you should consume more of or things that you should avoid. So that's one example.
Hmm. And I think it's fascinating because there's so many different pieces of machinery in the endocannabinoid system, and just what we know about the different splice variants of CB1. I was just looking over my notes today, and I saw one I forgot about. It causes a variation in gaze duration for happy faces. So depending on the type of CB1 receptor you have, you look at a happy face longer.
Yeah, no, you're absolutely right. It's fascinating. There's so many different variables to this, but it doesn't mean just because you have a propensity towards that, it doesn't mean it's going to express itself. So I'm not going to throw a brand under the bus, but I've taken every single genetic test just to do my research. And one of them told me that I am a very light sleeper and I don't do well with caffeine. Well, guess what? You can fire a cannon next to me when I sleep. I'm not going to wake up. I'm a very deep sleeper. Also, I drink 5, 6 cups of coffee a day every day. It doesn't affect me. Now, to say this, just because you have a genetic propensity towards that, it doesn't mean it's going to express itself. So, what we look at and we try to gauge that accurately is you have a genetic propensity towards this. You can have environmental triggers that can trigger this. If you are consuming cannabinoids and terpenes that are associated with these possible triggers, you may experience that effect. So you may experience, more happiness. You may experience more anxiety. But the things that we really want to focus on when it comes to that is things that you really need to avoid. So I'll give you another example. There's a group of genes that are called CYP genes. So CYP2C9 is a gene that's associated with metabolism. So the example that I use is like lactate, right? So if you're lactose intolerant genetically, you can metabolize lactate through the digestive system. Well, some people are not able to metabolize THC through the digestive system. So guess what? If you're consuming an edible or you're consuming something that has to go through your digestive system and your liver, the enzymes will not be able to metabolize your THC. So you will have a very negative experience. So all you had to do was know this about yourself and say, I'm going to consume my cannabis sublingually, or I'm going to, which is under your tongue, or I'm going to consume that through my buccal cavity, which is in your cheek. And now you avoid your digestive system and you avoid having that possible negative experience.
I like that you focus on those negatives because one of the things that you see a lot in these association studies is propensities to dependence.
There are a number of genes that are associated with dependence. So cannabis specific dependence is yeah, there are definitely genes that we look at. So we flag those genes and we give you the assays that are associated with dependence. So some people are slow metabolizers, some people are fast metabolizers, and some people, when that gene is expressed, they can have more of a dependency. Their CB1 receptors tend to stay locked longer. And there are also people that have a propensity towards dependence on opioids, for instance. So we'll also look at those and show you that if you have a propensity towards an opioid dependence, perhaps you may want to avoid consuming or getting an opioid prescribed to you, or maybe you can share that with your healthcare professional and look at an alternative to opioids, being cannabis is one of them. So yeah, there are definitely some markers that you can look at that makes a person much more susceptible to possible dependence on cannabinoids as well, specifically THC.
Another question is, for all the people you talk to, what seems to surprise them the most out of the results that they would get from this report?
The surprise that I question I always get is the people always want a cure all. So the thing that people and especially people who are less informed about the endocannabinoid system, they want to know, hey, what is what is my formula? What should I take that'll take care of all my needs? And the answer is there is no one formula. You have to look at all your different genetic expressions and know that some things are good for certain times, some things are good for other times. So I'll give you an example. So a lot of people talk about CBD, and they talk about CBD as I'm suffering from pain. I'm going to consume CBD. Well, there's a lot of things to really know that depending on where your pain is associated with, that's really important to know what type of cannabinoid and terpene ratio is right for you. But I believe in using the entire plant, but just using different parts and ratios of that. So you have to understand what it is. What is the goal that you're trying to achieve? What's the root cause of the pain? What's the root cause of the insomnia? So if we can look at that a little bit and look at the expression of that, we can help guide people on things that are better for them at better times, different times. So there is no silver bullet. It's not a one size fits all. And you may want to look at all those different factors. And based on different situations and different needs, you may have different ratios and different formulations.
And that makes me think of a question on the difficulties of interpreting all the science, because these genome-wide association studies, they're notorious for being very hard to interpret, very hard to get really solid data off of. So what's it like for your company to be taking in all of this research coming out about cannabis and the associated genomes and trying to figure out which ones you think are really solid and which ones you're not as sure you want to, rest a lot on?
You're probably sitting in our science meetings, and that is the biggest challenge. It's so interesting because you have studies that say, okay, we have 10 men aged 30 to 35 who are Asian living in this part of Japan, and that's your study. Now, that's a very narrow study. Do we incorporate that or do we not incorporate that? Usually, we don't incorporate those types of studies. We look for essays and studies that are a lot broader than that, that have a much more general population. The more research is being done and the more clinical trials are being done, the more we can incorporate that research. But yeah, that makes it very, very difficult. The other thing that we have to do is also create something that has a summary that an average person can read, because these scientific documents are so in depth and have such language that is so confusing for me that I'm the average person is reading that is going to have a really difficult time. So we have a rule of thumb. A rule of thumb. We have a summary. That summary we call the Bob from Iowa summary. No offense to Bob from Iowa, but it's the average person. So we try to take that data that's really scientific and in depth, and we try to provide a little summary of what that assay, what that study, what that summary really means. And that way, somebody can engage and really look and see if we can interpret that in common nomenclature. And yeah, so that way we try to take those really, really difficult to understand studies, interpret them, figure out which ones make sense to add, then figure out which genes they're associated with, then figure out is there enough cannabinoid research or terpene research, and then write a summary that somebody can understand. Because we look at two primary cannabinoids and a dominant and secondary terpene. So we look at CBD, we look at THC, and we look at two terpenes. Now, there is a lot of information out there about other cannabinoids, THCV, CBDV, CBG, CBN, and a whole bunch of other terpenoids and flavonoids and all that. But there has not been enough peer reviewed research on a lot of them that we felt strong enough that we can add. And the second part of that, there hasn't been enough test results for the actual products that people can go and consume to associate that with a suggestion. So that's where we sort of kind of teeter on what's appropriate and not to get too far ahead of ourselves.
That summary has to be some of the hardest parts.
That is the hardest part because we have a couple of professors who major in English literature, and there are PhDs in that. And we go back and forth and really, really go through and edit. And then we give it to a small focus group and let them read through it and see if they understand what it means. And yeah, that's a big struggle for us to figure out how in depth can we make it and how simplified it needs to be. And the more simplified it is, the better somebody can get real effective results from that.
And so, away from the plant and into the humans, of the different endocannabinoid machinery, which do you think we have the strongest data for about how they work and how these different various variants and SNPs change things?
Yeah, I think we've done a lot of research on our metabolizers. I mean, we have a lot of the association with the pain genes, the AKT1s, the COMTs, but I think some of the best research that I've seen is on the metabolizer genes. So there's and the reason why is, and I feel that this is something that's going to be uh, communicated in a very near future. There's a lot of challenges with different drug interaction, and we are not at all flagging drug interaction. So the research that I've seen coming out on the P450 enzymes has been incredible. And that's what we're looking to see because you may be a fast metabolizer, you may be a slow metabolizer. The level of metabolism and what else you put in your body, I think that's where we're going to see a lot more research. And that's where I see a lot of the newer essays going away from medicinal conditions like medical conditions like epilepsy, autism. All those studies are amazing, but because we don't focus on medical conditions, we focus on symptoms. I think that's where we find the most value in the studies that we look at.
It's especially fascinating to me, the foie molecule that breaks down anandamide in the body. And I'm just looking at a list from a review paper I read of all the things they found associated with that: lower threat reactivity in the amygdala, higher reward, less anxiety, higher impulsivity, increased likelihood to THC, but reduced susceptibility to becoming THC dependent, associated with cocaine, heroin, alcohol, and methadone dependence, and cravings for marijuana, and less severe symptoms, and lower happiness after actually smoking marijuana. Um, it's an amazing array of traits to associate with just one enzyme breakdown enzyme. And yeah, I was just curious how you put all of these players, all these different genes, all these different players in the body, and try to make sense of them for the average person.
Man, you brought up one of the best ones. So Fa is an incredible. I mean, there's a couple of other ones like that, but Fa is incredible because Fa runs a gamut. So you can look at the different symptoms that we look at: anxiety, depression, sleep, pain, all those different factors. And Fa is horizontal, runs across all of them. So it's very, very interesting to look at which specific RSID in Fa, where it is on a chromosome, what your genetic profile, what your genotype is on that allele, and how it affects that specific symptom. So I'll give you one more example on that. So I was talking about the NFL player, for instance. So one of the things that we flagged, there was it was a homozygous genotype, which means you're getting the exact same chromosome from the parents, homozygous genotype on a specific marker in Fa. And that had to do with anxiety. But specifically, it said, and I'm paraphrasing, but it said that this person will experience less anxiety in a threatening situation than other people in that sort of genotype in that population. And I asked this person live in the air, I said, does this seem correct to you that, they experienced that? And he said, basically, as a team captain and as a signal caller, and, this was the quote, when shit goes down, I feel very calm. So it actually works in that way. They feel very calm when anxiety is at a heightened level. However, the flip side of that, and that's the other end of FA, it said that this person also may experience, so this person experiences anxiety when things are calm, they actually work really well when there's a heightened state of alertness or it's a heightened state of anxiety. They don't feel that anxiety like, the person, a person in the average population. So it works really well. On the flip side of that, they had a propensity towards having a very, very negative reaction to opioids. So I read, you can experience these types of symptoms, dependence, and all kinds of different symptoms associated with that. And that person basically came back and said that throughout their NFL career, they got extremely ill when they consumed an opioid and did very, very well with cannabinoids and a couple other substances as well. So every single time we were pretty right on. Now it's not 100% of the time because it may not be expressed that way, but your FA gene is a huge indicator, and I think we're just at the tip of the iceberg to see how it actually interacts with the endocannabinoid system.
To something a little bit more technical, I was curious about the actual testing of the DNA. So you simply take a swab from someone's cheek, and then you have your own custom designed chips looking for endocannabinoid SNPs. And I was wondering if you would define SNPs for people, as well as just how you actually do this sequencing, especially of all the people who must be coming through.
Yeah, I mean, basically, so a SNP is a single nucleotide polymorphism. And how it works is it actually focuses on a part of your DNA strand. So within your DNA strand, you have your SNPs, which your polymorphisms within your nucleotide. Within that, you have an RSID, which shows you exactly where it's located on your chromosome. And within that, you have your allele combination, which is your genotype, which is specific to you. How our test works is you have a direct to consumer kit. It's actually a self-contained box. That box comes with it's a buccal swab, so it's like a giant Q-tip in a little tube. So you take the Q-tip, you rub the inside of your mouth where your buccal cavity, so inside your cheeks, basically. You try not to eat or drink for 30 minutes before. You put it back in the tube, you close it, and you put it in the bio bag that it comes in, and you close the box and you mail it. It's already self-addressed stamp. You mail it to our lab. And you're absolutely right. We have a specific array that we focus on, which has our SNPs on there. And when the array goes to your DNA, and by the way, there's a little bit of liquid, uh, which maintains your DNA, so it keeps it suspended until it gets to the lab. And then from there, we put that into a sequencer and we sequence your DNA and we see which genetic marker, which polymorphism you have. And then we report that in through our portal. We translate that and we provide you the results basically. So it works pretty much like that.
It's fascinating, new type of science. And that's why I enjoy your company motto so much. Cannabis is personal.
That's exactly the case. Cannabis is personal, and we all have a personal experience with cannabis. And hopefully, by doing an Endocanna DNA kit, you can have a personally positive experience.
And so before I let you go and get back to work, what's coming up that's exciting for Endocanna Health and your own work?
So we have a few clinical trials that we're going to be starting in Australia. So the clinical trials are standardizing on our kit. So every single person is going to go through that trial is going to have to take their genetic test. So we're going to be able to hone in a little bit more, not only in an endocannabinoid profile, but how that endocannabinoid profile works with a specific condition with a specific genetic profile. That's very, very exciting. The second thing that we're going to be focusing on is associating different types of reports that focus with your cannabinoid experience. So something about nutrition. How does your nutrition also interact with your endocannabinoid system and possibly expression of those genes? And a couple other reports that are similar to that. So we're expanding our library reports. We also are finishing our matching engine, and the matching engine will basically take products that are geo-tracked, so anything that within your zip code will be able to match those products with our suggested ratios and show you here's 5 different products that are very close to 100%, maybe 50% of what our suggested ratio is, and here's where you can get that product. So you don't have to really even go through with your sort of menu and go to a bud tender and ask, do they have this? But it'll just have these brands and products depending on the state that you reside in. Make sure it's compliant with that, but where you can actually get that specific product.
Wow. So from your genes to your front door.
That's it. Well, from your genes to the front door, because there will also be some delivery services. So from your genes, you click on, you get that alignment, you click a button, and you get it delivered to your door. How you like that?
It's a brave new world.
That it is. It's great. I mean, coming from knowing so many people that spent so much time in jail for cannabis, having all kinds of legal difficulties, all kinds of issues, and the stigma that's associated. Hey, I went through it myself. My parents asked me to leave my house when I was a kid, when I was a teenager, basically, because they didn't believe in this as my own medicine. So so now the irony of that, I actually suggest and recommend and help make certain formulations for my parents to help with their conditions. It's such irony to come full circle and have that conversation with my dad. My dad said, "You know what? We're brainwashed. We didn't know until we experienced it ourselves and took away the stigma. And now the stigma is gone. They're actually enjoying the benefits that cannabis can provide for them."
Oh, that must be a good feeling.
It's incredible. It's an incredible feeling to be able to have something that I've done for so long and be my passion now to come to fruition. And the more people we can help, the more stigma we can remove. And this is going to become normal. Like I was talking to my daughter about this, and she's 13, almost 14. And to think that she's going to be with her friends at some point when she's my age or maybe a little bit younger, and she'll be like, "You guys actually people went to jail for a plant?" It's going to be insane for them to even believe that. But that's exactly what it was. People go to jail for a plant.
Well, thank you so much for your work and for taking the time to talk to us today. And best of luck with Endocanna Health.
Thank you for having me. I really, really appreciate it.
Greener Grass is a Bluebird Botanicals podcast. Their CBD oil supports a healthy body and a strong endocannabinoid system. They've got friendly customer service who can answer any of your questions, and the number is right there at the top of their webpage. But, per the FDA, they won't be able to make any medical claims for these nutritional supplements. That's also the reason you'll hear little about CBD on this show. There's no need for us to add more evidence about CBD when a simple Google search will give you more than you can read in a month of Sundays. So this show covers the cannabis world and more with editorial freedom from Bluebird Botanicals. Thanks for joining the Greener Grass podcast. As always, our audio alchemist is Matt Payne. The Gypsy Jazz theme music comes from Brett Van Donsel. Our beautiful bird sounds are courtesy of Lang Elliott. And I'm your host, Lex Pelger, wishing you a bright green day.